Moll's half-life experiment. Moll's half-life experiment is popularly known to be performed to demonstrate the necessity of carbon dioxide for the process of photosynthesis to form starch. Photosynthesis is a vital biosynthesis process where chloroplast of green plant synthesize sugar by using water, carbon dioxide in the presence of sunlight. The glucose which is produced through the photosynthesis is later converted into reserve food material, that is the starch. Material required: a de-starched potted plant, caustic potash, wide mouthed bottle, iodine solution, split cork and water bath. Take a de-starched potted plant. A de-starched potted plant is a plant which is kept under dark for 2 to 3 days, where photosynthesis is inhibited and the reserve food material, that is the starch, which are present in the plant are completely utilized for the metabolic activities. And a wide mouth bottle which contain the KOH pellets. And a split cork. Make KOH solution using the KOH pellet by dissolving it using distilled water. Or you can also pre-prepare the solution and keep it as a stock. Now open the split cork and place the leaves in between the two halves of the cork of the bottle, so that the half the leaf remain within the bottle and the other half outside the bottle. The apparatus is made airtight by applying melted wax or grease, so that the atmospheric air may not enter the bottle and disturb the setup. Thereafter, the apparatus is kept in the sunlight for the photosynthesis for 4 to 5 hours. After a few hours, the leaf is removed from the plant and it is tested for the starch by extracting its chlorophyll and keeping it in iodine solution. Test for starch. Take the leaf and insert it in the ethyl alcohol. The test tube in a boiling water bath. Avoid the boiling of the test tube in the direct flame. Keep it under the boiling water bath for a few minutes. After the few minutes, take the test tube out and you can see the change in color of the alcohol solution. It turns into green color, which indicate the green color pigment, that is the chloroplast, are extracted from the leaves. Separate the portion of the ethyl alcohol solution or discard it. The change in color of the leaf from green to colorless. In the test tube, green color, which contained the ethyl alcohol along with the green color pigment, and the leaf lost all its pigment. Now wash the leaf with some water. Now test the leaf for the presence of starch by adding few drops of iodine solution and leave it for a few minutes without disturbing it. After a few minutes, you can observe that half the portion of the leaves are turned to bluish black and remaining portions are still colorless. In the leaves, the base portion, which receive all the necessary factors which are required for the photosynthesis, the carbon dioxide and the water and the sunlight. Therefore, the photosynthesis will take place and it will produce the glucose. Later it is converted into starch and it gives the positive test for the starch. Whereas the remaining portion, that is the tip and the middle portion, where either carbon dioxide is miss or absent or the light, therefore they give negative for the starch formation test. From the above experiment, we can conclude that the rate of photosynthesis is dependent on its limiting factor, that is the light, carbon dioxide or water. If photosynthesis is to happen at its maximum rate, all the environmental factors must be present at its optimal level. If one or more factors are in short supply, the rate of photosynthesis will be limited.